Welcome back to the Classics of Immunology Journal Club. And today we are in 1995. And it turns out that 95 was a big year in immunology. We're going to be reviewing a paper from the Journal of Immunology. And before we get started, don't forget to go on the YouTube channel and like the video and click on the bell and to get notified for more, and check out the website and look at the writing tab and read this particular paper. This paper requires a little bit of study. And so I recommend it highly. And the paper is by Shimon Sakaguchi and his wife Noriko Sakaguchi and three other Japanese authors. And it's entitled "The Immunologic Self-Tolerance Maintained by Activated T Cells Expressing the IL-2 Receptor Alpha Chains," in parentheses CD25, which I will talk about. It's published in the Journal of Immunology in 1995. And the background in this for this paper is that Shimon, his wife was a dermatologist. And actually, she saw me and took care of me at one of the international immunology meetings that happened to be in Kobe, Japan, a few years back. This paper, in terms of the background of this, goes back to the 1960s, 61, 62, and to Jacques Miller. I don't know if you remember, we reviewed that, one of Jacques Miller's papers, where he established essentially that the thymus was important for immunology, and it was important for the maturation of and the generation of peripheral T cells. That's why, because they come from the thymus, that's why we call them T cells. And what Jacques Miller did back in those days is that when they had thymectomized adult mice, put them back in the cage, they were just fine. They nothing happened to them, and so they couldn't figure out whether or not the thymus was important or not. Jacques Miller went the extra mile, and he devised a method to thymectomize newborn mice, which are just tiny little things. And if he worked out a system where he thymectomized them within the first 3 days of life. So it was day 3 thymectomized mice. And when he did that, he found that when he put them back in the cage, and he also did sham thymectomies on litter mates, that they looked fine for the first month of life, and they grew normally, and they seemed to be okay. But then in the second month of life, they started to become wasted. They lost weight. They became runted. They started to develop all kinds of auto-inflammatory symptoms and signs. And essentially, Jacques Miller was the first to establish that if you thymectomize these mice, that he went on to do other experiments, and so he showed that they were immunodeficient, which is what he was hoping for. But in addition, in the second month of life, they became autoimmune. So you had this paradox of immunodeficiency and hyperactivity autoimmunity on the other hand. That was he focused on the first month of life and the immunodeficiency. It was never really explained and experimented upon in that second month of life. In the second month, there was an infiltration of what looked like activated lymphocytes in all kinds of organs. And then the mice would succumb and they would die. It was very much like graft versus host disease. So nothing happened for 20 years until 1982. And Shimon Sakaguchi, now this paper I'm going to review today is from 95. In 1982, Sakaguchi demonstrated that after thymectomy in newborn mice, day 3 thymectomized mice, just like Jacques Miller had done, that these mice would develop okay for a while, and then they would develop all kinds of autoimmune diseases, multiple autoimmune diseases, thyroiditis, oophouritis, gastritis, all kinds of things were going wrong. And he found that if he would try to rescue these mice with normal T cells, that he could do that. He could rescue them. They wouldn't become autoimmune. However, if he took away what he thought was the helper T cell compartment, the LYT1 positive cells, and only gave them the rest of the lymphocytes, that then they would get this autoimmune phenomenon going on. He showed then in an accompanying paper in 1982, and these were out, I think, in the Journal of Experimental Medicine, that he could add back the LYT1 cells, which he thought were the helper T cells, and then that would prevent the onset of this disease, this autoimmune phenomenon. And then between 85 and 92, several other groups in the world had published similar kinds of papers, similar phenomenon that he references in this 95 paper. In 1985 and thereafter, Sakaguchi tried to pin down exactly. I forgot to say that in 1980, even though it was sort of gospel that the LYT1 cells marked the helper T cell compartment, Leonard Herzenberg from Stanford on the West Coast showed that if he used flow cytometry, he could show that all T cells had LYT1 of varying degrees, and therefore it wasn't a subset marker of only helper T cells. LY23, by comparison, did mark only a subset of cells, and these were ultimately became the CD8 positive T cells. But for years, the human people were interested in human lymphocytes, particularly my good friend Alice Reinhardt had shown that there was a marker, a monoclonal antibody that marked a molecule called CD4, and that's what Sakaguchi thought he was dealing with, but he wasn't. Anyway, as time went on, when we developed the IL-2 receptor assay in 1981, it would give us data on the total cell population that whatever we were trying to find out about. And so when we looked at resting lymphocytes, we could detect a small amount of radiolabeled IL-2 binding onto resting lymphocytes. And for the most part, we looked at human cells, but we also looked at mouse cells as well, spleen cells, lymph node cells, and so forth. And it wasn't until, that was 1981, it wasn't until 1990, a decade later, that the antibodies had been developed that would mark the alpha chain of the IL-2 receptor and the beta chain of the IL-2 receptor. The gamma chain still had not been discovered yet until 1992. But in 1990, we re-looked at the expression of IL-2 receptor alpha chain and beta chain on resting peripheral blood lymphocytes, because we were interested in using IL-2 in the clinic to see if we could generate large numbers of either antigen-activated T cells that might be there in a small proportion, but also we were interested in looking at natural killer cells. And we found when we did that if we looked at CD4 and IL-2 receptor alpha chain, about 5% of the CD4 positive cells in a normal volunteer were positive for the alpha chain. By comparison, only about less than 1% of the CD8 positive cells were positive for the alpha chain, I'm sorry. And we didn't find beta chain on either one of those subpopulations, CD4 or CD8, which is of interest. So in the introduction of this paper, Sakaguchi hypothesized in the very first paragraph of this paper, and he reiterated the data that were already out there in the literature, that there were various autoimmune diseases, either organ-specific or systemic, like lupus erythematosus, for example, or rheumatoid arthritis. He hypothesized that these might be due to a breakdown of a common mechanism required for self-tolerance, and essentially for peripheral tolerance, but required for self-tolerance. That was his working hypothesis based on his 82 papers and 83, 4, 5, and so forth. And he also hypothesized that it was the T helper cells that were responsible for the autoimmune diseases. He also pointed out that he could reproduce these diseases by removal of particular T cell subsets. One of them was CD5, and that would mark a subpopulation of CD4 positive cells. And if he took those out, those cells now would result in the autoimmune diseases. So he concluded that the potentially pathogenic CD4 positive cells were regulated by another subset of CD4 positive cells. And he was searching for a more specific marker than LYT1 or CD5 that would recognize whatever this so-called, he hypothesized in 1982, and he called them regulatory T cells at this stage of the game. And that's what this whole thing is all about. And so he looked at data that were out there in the literature and so forth, and he keyed in on the IL-2 receptor alpha chain. And he thought, because this was on a very small, much smaller subfraction of cells, around 5 to 10% in the mouse, in the peripheral cells, either coming from the spleen or from the lymph node, which were the two most readily available organs. That was it at that particular point in time. And in terms of the materials and methods in this paper, he focused on BALB-C mice, nudes, or their heterozygous littermates, nude plus, and he would take cell populations from these animals, from the heterozygotes and also from the nude, and he would mark these and or use complement to remove subpopulations from these from the different organs. He focused on CD25, IL-2 receptor alpha chain, CD4. He had an antibody to that Frank Fitch had generated, and he had LYT2.2 antibodies that would mark CD8 positive cells, and he had anti-CD5, and he would incubate different populations of spleen cells and lymph node cells with the antibodies plus complement at 4 degrees for about 45 minutes. And then he would add complement, which was rabbit serum, and lyse the cells that were antibody positive. And then he would inject the mice with these cells intravenously. He also used flow cytometry and also the fluorescence activated cell sorter to sort different populations. He also looked at the clinical histologist, histologic analysis of the different organs that he was interested in to see whether or not they had evidence of inflammation in the organs. He also bled the mice and tested for antibodies using ELISAs to different autoantigens as well as non-self antigens. And we'll go over those data. So the results, and now we should do some screen sharing. And this is figure 1A. And in this figure, we have the expression of the alpha chain on the x-axis, and we have different submarkers on the ordinates, and these are flow cytometry plots that hopefully you all are familiar with. The expression of the alpha chain on mouse peripheral T cells, and these were 2-month-old mice, and so they were clearly of adults, and you can see that. So this is CD3 versus IL-2 receptor alpha, the alpha chain, CD4 against the alpha chain, and CD8 against the alpha chain. It's interesting because CD3, there's a certain fraction, about 10% of CD3 positive cells are alpha positive. And again, CD4 about 10% in this is just one experiment. CD8's not so much, 0.1 out of like 17.5% here. And whereas the CD8 negative cells are alpha chain positive. Here's CD5 down here to give you an idea what's going on here. So about 10% of CD5 positive cells are also alpha chain positive. They are CD5, 45 RB negative. And here we're looking at cells with just the beta chain versus the alpha chain. And you can see that they're only about 2% of the total cells from the spleen, I think this is, are positive for the beta chain, and about 7% are positive for the alpha chain. And these are probably NK cells here, because clearly the CD8 positive cells are not beta chain positive. now that's contrary to subsequent data coming out of John Sprent's lab, where he felt that the memory CD8 positive T cells were beta chain positive because they had IL-15 receptors. So the next thing we want to look at here is table 1. And I usually don't like to do tables, but this is how the data have been presented in this paper. So you have to study this a little bit. This is the induction of autoimmune disease in nude mice by inoculating nude plus T cell subpopulations. And so, and if you look here, the experimental group here, A through H, and then the treatment of the cells, total number of mice examined, and the number of mice with autoimmune disease. And so in the first, in the A group, where we have, we just treated the cells that were injected into the nude mice with complement, so that's a negative control, and nobody gets autoimmune disease. And here we are, and B is exactly the same thing. We have the treatment of the cells was lower, and we don't have any autoimmune disease. Now in C, D, and E, we have different doses of IL-2 receptor positive or antibody, anti-IL-2 receptor plus complement, 10 to the 8th cells, 5 times 10 to the 7th cells, 1 times 10 to the 7th cells. And this is the, if you just look at the percentages here of the number of mice with autoimmune diseases, at the highest cell concentration, we have the most highest number of or percentage of cells or of mice that develop the autoimmune disease. And as we go down in cell dose, we get fewer. Then these are other controls in this experiment. The B cells, if they injected B cells or macrophages or monocytes, there really was no effect whatsoever. Now, the other thing, and so clearly a lot was going on here from a biological standpoint. Now, if you look at the evidence of autoimmune diseases in these mice, by looking at antibodies, we've got several different antibodies we're looking at. In the upper panels here, this is anti-parietal cell from the stomach antibodies. This in this panel is anti-thyroglobulin, thyroiditis antibodies. And you can see that the mice that received cells that were, the alpha chain was removed, have a very high antibody titers. Here's up to 40,000 here. And also the thyroglobulin titers, whereas the non-treated cells are perfectly fine. Now, if you then look at so that's organ-specific kinds of problems. There was also systemic autoimmune evidence, and here's anti-double-stranded DNA antibodies. And again, here we are, the treated population, then the non-treated, and also non-self antigens. This is very interesting and very important given what happened in the history of immunology ongoing, because here we have anti-TNP antibody, and those are positive versus the negative controls. There was also diffuse hypersplenomegaly. You can see that here. And hyperimmunoglobulinemia, and you can see that here. And so these mice had evidence of both systemic as well as organ-specific autoimmune phenomena going on. And that's all I really want to show at this stage of the game in this. There's other data that are supportive data, and you should go read the paper carefully and look at the figures and the tables. So let's move on to the discussion. The discussion of this paper is also very interesting because he's been in this mode of trying to sort this phenomenon out now for at least 15 years when this paper came out in 1995. And so they make the following points in the discussion. The first is that the expression of the IL-2 receptor alpha chain could divide the normal peripheral CD4 positive T cells into potential self-reactive, that would be the CD5 or the alpha chain negative cells, and another which he called controller cells or regulatory cells, which would be alpha chain positive. And so he brings up right away in the discussion, the question is, since we know already that the alpha chain marks recently activated antigen activated cells, is this regulatory cell phenomenon the same thing as the activated cells, or are they separable? Now, we know now, after many more years, that they are separable. And I'll go into a little bit about that, but not all. But at that time, you couldn't separate them. Phenotypically, they looked identical, these two subpopulations of CD4 positive cells. He also speculates or conjectures in the discussion that these IL-2 receptor positive cells, they might act on non-T cells, in addition to the so-called effector T cells that arise. And he speculated that maybe the APCs are important in this whole reaction. And that becomes also important as history moves on. The other thing is, because of the fact that this marker seemed to be so specific for these regulatory or controller T cells, that they might simply be binding IL-2 and act as a means of depleting IL-2. And of course, my lab particularly and other labs had shown that the IL-2 receptor, when it binds IL-2, that the IL-2 receptor complex is rapidly internalized, and the alpha chain is recirculated to the surface. The beta and then the gamma chain are degraded along with IL-2. So what happens is that you will deplete IL-2. The IL-2 receptor positive cells will deplete IL-2 in the surrounding milieu. So that could be sort of acting as a cytokine sink and just removing IL-2. And that's interesting. The other thing he said, he's trying to understand the mechanism, or is postulating what kinds of mechanisms might be involved in this whole process. And he says, these cells might secrete suppressive cytokines that are known to be dampen down the immune response. And in particular, IL-10 was conjectured early on, together with TGF-beta. And those two cytokines got a lot of play subsequently in the literature. And the other thing he points out, and this was sort of, the people interested in autoimmunity really latched onto this whole phenomenon. And whereas the people that might be interested in just regular immune reactions to non-self antigens, foreign antigens, he pointed out in this paper in 1995 that the regulatory T cells in a normal situation might also dampen down the immune reaction to non-self antigens, which is very important from the standpoint of potential therapeutic uses of regulatory T cells in cancer and also infectious diseases, if you wanted to get rid of them so that you could have a better immune response. The other thing he points out, or he conjectures about, is the fact that it's quite possible that various autoimmune diseases that we see in the clinic, in the human, are not due to total depletion of the regulatory T cells, but just a minor diminution in vivo in terms of maintaining the balance of the self-reactive immune mechanism. So that's it in terms of this paper. I recommend you go back, you read it carefully. This paper is really a wonderful example of why it's important if you happen to be a student of immunology, and if you happen to have ambitions to be an immunologist that stays in the business for your entire career, it's really important to understand the history of how things developed. And that's why I went into the situation with Jacques Miller in 1961, and Sakaguchi's 82 paper, now this 95 paper. The 95 paper was pivotal because it pinned down the IL-2 receptor alpha chain as being the marker, which has persisted then for years thereafter. This is a way you would recognize Tregs. The other thing and so that was good, but the thing that I was sort of disappointed about was the fact, and again, the autoimmune people, everybody keyed on the fact that the other name for IL-2 receptor alpha chain is CD25. So people began to talk about CD25 as if it was just another marker, ignoring the fact that, well, interleukin-2 could very well be very important, and its receptor could be very important for the generation of an immune response. And so if you wanted to regulate that, maybe you would be regulating the IL-2 molecule itself and or be regulating the expression and the function of the receptor, the IL-2 receptor, which is what happened. People just sort of ignored the fact that it was might be a functional importance. It was just a marker for Tregs, the CD25, to a lot of people. My lab didn't get involved in this early on. I sat back and watched it for several years, but you know, I was always sort of bothered by the fact that when back in the days when we developed the IL-2 receptor binding assay, radiolabeled binding assay, Alan Monk, my guru, my endocrine guru, who taught me all I needed to know about endocrine receptors, said that if it's a true hormone, there is a negative feedback regulatory control over the hormone. Otherwise, once it's set in motion in a positive way, it will just continue on and on and on. And now that wouldn't be so good for T-cell leukemia or lymphoma. It could just be that you needed to turn off the IL-2 mechanism. But we never identified a regulatory feedback negative mechanism. Turns out that this is the mechanism. And subsequent papers in this business that we wrote and came out of my lab approached the problem from that standpoint. Other things that happened subsequently after this 95 paper was in 98, Ethan Shivak. The problem with Sakaguchi's setup was that it was in vivo, very difficult to manipulate. He was good at it because he'd worked on it for 15 years, trying to find the subset of cells that were doing the deal. In 98, Ethan Shivak at the NIH developed an in vitro assay with taking just regular spleen cells, activating them in vitro with anti-CD3, and then adding in his experimental system, the putative regulatory cells, and then reading the readout of this assay was proliferation. And again, since proliferation is dependent upon interleukin-2 primarily, maybe not 100%, but 95+% essentially, I felt, and what he would do is he would titer in the regulatory population, and they would dampen down the proliferative response of the effector T cells, but he needed a lot of them. He needed at least 50% of the cells in the culture to be the regulatory cells, and when he titered them out to less than 10%, the regulatory phenomenon went away, was gone. And I thought that this was confirmation that at least one of the mechanisms whereby regulatory T cells worked, which is what Sakaguchi speculated in this particular paper, was as a cytokine sink. It was just sucking out all the interleukin-2. And I had a paper out with Gregoire Altan-Bonnet in I think 2010, where we looked at that very, very carefully, and I recommend you read that paper too. So what happened subsequently is that then in 2001, the scurfy mouse mutation and in the mouse and the mutation in the human that led to both systemic as well as organ-specific autoimmune disease. The XLAAD phenomenon was found to be due to a mutation in a gene that coded for FOXP3. And that became subsequently very important. And then between 2001 and now, there are many papers that we will get into as time goes on, looking at the molecular mechanism of how these regulatory T cells actually work. One of which has been shown very carefully by Pushpa Pandian and Mike Leonardo at the NIH to be due to the removal of IL-2 in the whole thing. But that's not the total story. There's other molecules involved, such as CTLA-4 and PD-1 and so forth, that are very important subsequently as we get into this whole process. So I recommend that you check those out too. So that's it. Don't forget to like the video if you do, and click on the bell and check out the website, check out my Twitter feed, and come back next week. Thanks. The other thing that's very important is that if you really want to get a synopsis of this particular field, a corner of the field, you should buy my book, Molecular Immunity, because in this book is a very nice, if I don't say so myself, discourse on regulatory T cells in chapter 8. And this really puts it all together for you. If you read this chapter, which is not too long actually, then I think you'll see the light. So thanks again.